Okay.
Here for.
So phoenixin was identified about 3 years ago by Liu and Yostin and other colleagues, and they found that phoenixin acts in the reproductive axis. So this incorporates the hypothalamus, the pituitary, and the gonads. To briefly go over some background, so GnRH from the hypothalamus stimulates gonadotrophin, so LH and FSH production from the pituitary, which then stimulates sex steroid production from the gonads. So after detecting phoenixin in the pituitary and discrete regions of the hypothalamus, Liu and colleagues were interested to see whether phoenixin would affect pituitary hormone secretion. So they decided to apply phoenixin in vivo to in vitro to rat pituitary cell cultures. So what they found was that anterior pituitary hormone secretions were not affected, but in the presence of GnRH, there was an enhanced LH secretion. So this corresponds with further findings in the next graph to the right, showing that phoenixin was able to stimulate GnRH receptor mRNA in the pituitary. So strengthening phoenixin's role in the reproductive axis, they applied specific small interfering RNA. These are molecules used to inhibit the production of phoenixin, and this ultimately lengthened the time between signs of estrus or signs of heat in the rat, suggesting phoenixin's importance in maintaining cyclicity. Studying another animal model, such as the chicken, can help us further elucidate phoenixin's roles in the reproductive axis. So, particularly with an immature bird and a fully reproductive mature bird who is laying just about every day. At the level of the pituitary, we're still unsure what regulates receptor 3 expression. We know that gonadotrophin releasing hormone, GnRH, itself partially regulates receptor expression, and it's possible that estrogen and progesterone in turn feed back onto the pituitary and regulate GnRH receptor, but we're still unsure. So it gets down to my hypothesis here that phoenixin is a candidate for regulating gonadotrophin releasing hormone receptors expression. So what we wanted to do first is we wanted to identify phoenixin in the chicken genome. So we ran the identified peptide from the original paper into online databases such as NCBI, UCSC, and Ensemble. And we were able to find that the peptide was just about over 80% identical to the chicken from the rat. From there on, we wanted to see if the mRNA was expressed in different tissues. So this means that the DNA is transcribed, and it's possible that the mRNA is translated into protein. So we dissected tissues such as the diencephalon, which incorporates part of the hypothalamus and the brainstem, the pituitary, the ovary, the gut, and the heart. And we took RNA from this, reverse transcribed it into cDNA for greater sample stability, and we ran a polymerase chain reaction and parallel with beta-actin as our housekeeping gene to control for sample integrity. And we found that phoenixin was expressed in chickens over 15 and 29 weeks of age. Those are the numbers that are expressed there under each tissue. So to further quantify phoenixin's expression, we ran a semi-quantitative PCR to determine if there's differences in between the ages. So phoenixin was significantly expressed in the hypothalamus, or diencephalon, at 15 weeks of age. And we also found that phoenixin was expressed significantly in the gut at 29 weeks of age. So phoenixin has a wide expression panel here, and it was similar to the rat, suggesting that it may have similar function. So phoenexin could possibly play a role in reproduction, potentially in the beginning when the chickens are sexually maturing. Unfortunately, the pool samples that we took were from 15 weeks of age birds. They already started laying at 17 weeks of age before photostimulation at 18 weeks of age. So phoenixin could have already been priming the reproductive axis prior to lay. And it could also play a role in the gut at 29 weeks of age, because as the birds sexually mature, they have greater energy requirements. So in the future, we'll look at phoenixin's function in the chicken and see if it actually affects gonadotrophin releasing hormone receptors expression.
Okay, thank you. That's good. I know there is so much to talk about.